This is your instructor Kwame Shen, and part 3 of our topic, Introduction to Microbiology and Microscopy. We'll look at microbes and human welfare. And so if we look at microorganisms generally, scientists have studied different microorganisms, and depending on which microorganism they're studying, they are described variously. So you have bacteriologists, mycologists, parasitologists, and biologists based on what they're studying. So bacteriologists will study bacteria, and with the advent of crude microscopes and the discovery of microorganisms by van Leeuwenhoek, gave birth to the whole field of bacteriology. When Pasteur and some of these other scientists studied bacteria and saw their role in the food industry, the environment, and the fact that they could cause certain diseases as well. And so that's a field that continues to expand. New discoveries are made every day. But bacteria may exist as single cells, or they may exist in groups by attaching to other bacteria and forming what we call a bioform. And when bacteria exist in a bioform, they are more resilient to treatment. So in terms of medical implants and hospital-acquired infections and the likes, you have all these bacteria that exist in the form of biofilms, and the biofilms gives them more protection from harmful environmental agents such as disinfectants that are applied to kill them. It's almost operates like a multicellular organism, the way the biofilms work. So if you should lick the sides of your teeth right now, you will feel a very thin layer, and that's a biofilm, and that's why we keep taking care of our teeth to prevent it developing into a plague. The study of fungi is mycology, and fungal infections are very important, especially in the immunocompromised, and so it accounts for about 10% of hospital-acquired infections currently. And with the increasing climate changes that are ongoing, we have seen an increase in the number of fungal infections as well. Then there's parasitology, the study of protozoa and parasitic worms. And so the study of malaria, Plasmodium, which is a parasite, will fall under parasitology. It's a study that is more relevant, especially to people in the tropics, but every now and then it becomes important even in the developed world. For a very long time, scientists were just aware of bacteria until 1892, when Dmitry Ivanovsky reported that the filter that they were using to filter out all known bacteria at the time couldn't filter out certain microorganisms which were causing mosaic disease in tobacco. In 1935, Wendell Stanley was able to demonstrate that organism which Dmitry Ivanovsky alluded to is the tobacco mosaic virus, the TMV. He realized that it was significantly different, very simple, and almost can actually be crystallized. So it had a very different property in terms of its characteristics compared to bacteria. And so that led to the discovery of viruses. And then with the advent of electron microscopes, now we can study viruses into even more details. So in terms of our warfare, vis-a-vis microorganisms or microbes, we actually benefit a lot more than we suffer from the effects of microorganisms. Because without microorganisms, we wouldn't have a clean air, our waste wouldn't be degraded, wouldn't have access to certain food like plants that would rely on nitrogen fixation and the like. So they are not really the bad guys. You have algae and cyanobacteria that would produce oxygen through the process of photosynthesis and carbohydrates, which becomes the basis of the food chain in the oceans. You have bacteria that help recycle important elements like nitrates and sulfur and phosphorus and the likes, to return nitrogen back into the soil and fix them in plants, and that it can become available to animals as well. Then we have also used them in cleaning the environment. Our sewage system, water treatment plants, and sewage treatment systems employ these microorganisms to help degrade the organic components of the waste. The reason being that if we don't do that and we just release these untreated organic molecules into our water systems, waterways and the likes, what's going to happen is that there's going to be eutrophication, and eventually it's going to destabilize the ecosystem. So they are very beneficial to us. We have used them or their products, so either the bacteria or some enzymes that they synthesize in cleaning out certain toxic wastes such as oil and mercury. During the 2010 Deepwater Horizon oil spillage in the Gulf of Mexico, we saw an increase in bacteria that actually use these hydrocarbons as a source of energy. And so we're able to clean the environment using these bacteria. We use bacteria, microorganisms, in pest control, biocontrol, where we have used Bacillus thuringiensis, a bacteria, to infect so many insects. And so we're able to destroy them. But this bacteria is harmless to other animals and humans and even to the plants. We continue to use microorganisms in biotechnology and producing food, the beverage industry. We've used biotechnology to produce insulin, which we use in therapy, the human growth hormone, interleukin-2, and we use these microorganisms. We use their plasmids, use restrictive enzymes to cut off and insert our gene of interest, mix them up, and allow it to just multiply. And in the process, we're able to have more of the products of interest that is of interest to us. We also have normal microbiota, the trillions of microorganisms that live in and on our bodies. We used to call these microorganisms, these bacteria, flora, but now the correct term is microbiota. So we have normal microbiota that lives in or on our bodies. They may be transient, that is, they live there for a very short period of time, or they may be permanent in or on our bodies, depending on the conditions and the relationship that we have with them. But the benefits that we have from normal microbiota is enormous. We benefit from growth factors such as folic acid and vitamin K that are produced by these microorganisms. They prime our immune system, and so they prevent us from getting infected. They also compete with potentially harmful microorganisms, And so because they compete with these potentially microorganisms for space and food, they're able to outcompete them. So we don't end up suffering from the effects of these pathogenic species. For a disease to be present in a host, such as a human, the host's resistance has to be overcome. And for that to happen, you should have to consider these three factors: the environmental exposure, the agents that is infecting the host, and how susceptible the host is, how healthy the host is in terms of the genetics, in terms of its nutritional status. And then the environmental exposure, you're talking about access to healthcare, social environment, emotional health, and the likes. And then you have the agent. Is it a virus? Is it a bacteria? Is it a prion? So whatever is the source or the agent, they all have to come together in a certain manner for the disease to present. So if there's an infectious agent and is not able to attach itself, there's no disease. If there's an infectious agent that is able to enter the host's body, but the host has very good nutritional status and very resilient in terms of its genetics and the likes, then the host may be able to fight off the disease. And then there's access to healthcare. If you have good access to healthcare, you're able to do preventative medicine, protect yourself, then there is no way you would get it as well. So these factors are important. So the resistance is the ability of our body to fight off infections, potential infections. And we're able to do that because we have several barriers. We have physical barriers and we have cellular barriers and chemicals barriers within our bodies, part of our immune system that help us to fend off diseases. You have physical barriers in the skin and mucous membrane. So if you have a fever or a cold, you realize that you produce a lot of mucus because it is a way that your body is fighting the infection. And by bringing up these mucus or passing it down in the stool, you're removing a lot of these microorganisms from your body. We have the cellular part of it, the white blood cells. There's an inflammatory response, the chemical aspect of it as well. And so that is part of the immunology in terms of the study of our immune system and how we're able to defend ourselves. So resistance is important. But pathogenicity, that is the infectious agents, how pathogenic, to what extent can this microorganism cause a disease in a host? So you're looking at characteristics such as the proteins or the antigenic proteins, properties of the surface of the microorganism. If the microorganism produces toxins like some species of E. coli, the sugar toxins, then that makes it more pathogenic. Some microorganisms have capsules, and the capsules that they have makes them more pathogenic. Some have peptidoglycan, more peptidoglycan or less peptidoglycan. So you have a number of factors that makes an organism more pathogenic or less pathogenic. So they all don't cause diseases at the same rate. We are witnessing something right now in terms of COVID-19, which is a new infectious disease. So the question for our participation exercise for the week is this: why is there increase in the number of new diseases and an increase in the number of even existing diseases in terms of number of cases? Why do we have an increased number in emerging infectious diseases? Why is there an increase in the number of new diseases, new infectious diseases? So that's the question of participation. So post this question and then give a response. So you're going to write a question, write your response to it, and post it together on Blackboard. Okay. So I'll go through a number of infectious diseases that we have so far experienced and which continues to grow. We in 2012, 2014, that's why 2015, there was actually an outbreak of what we call Middle Eastern respiratory syndrome. So there's a virus that belongs to the common cold family, and it causes a respiratory infection. In 2015, there were 36 cases that was associated with one person. One person was able to spread to 36 people. It's believed that it started in bats and then went to camels, and then from camels to humans. We had a first reported case in 2012 in Saudi Arabia. Then in 2002, there is a severe acute respiratory syndrome, SARS, was first discovered in China. It was reported as atypical pneumonia. In 2012, there was a declared agent that it was declared an agent that could cause actually a severe threat to public health. And we continue. It's been a while since we've had cases of SARS, but it's something that we haven't had a cure for it yet, and we don't even have a vaccine for it yet. Then there is Avian Influenza A. Avian Influenza A virus is primarily limited to poultry, waterfowl, and poultry. But every now and then there are outbreaks in humans. So, for instance, in 2013, there was an outbreak that infected 131 people in China. So far, we haven't seen transmission, human-to-human transmission, being sustained. And so that's good news. But generally, how it's transmitted is that people who live in close proximity with poultry become infected by directly coming into contact with the infected fowl, touching them or their stools or surfaces that they have actually used. That's very common. So and then there is also aerosolized aspects of the transmission, where you have the flu virus becoming airborne and someone breathing it in and becoming infected. Viruses can move from animals to humans, but for that to happen, it has to have the right antigenic or surface proteins for it to be able to attach and cause an infection. In 2005, scientists were able to discover that the avian flu or influenza A had infected pigs. Now, once that determination was made, it was only a matter of time before it infected humans, because pigs and humans have a very similar biochemical structure in terms of our constitution. There is also a lot of resistance to antibiotics. So you have methicillin-resistant Staphylococcus aureus, MRSA, which is something that we continue to battle now. So we had penicillin resistance developing in the 50s. A derivative of penicillin, methicillin, also becoming bacteria become resistant to it in the 80s. In the 90s, we have methicillin-resistant Staph aureus that have also become resistant to vancomycin. And so you have vancomycin intermediate resistant Staph aureus and vancomycin resistant Staph aureus. West Nile is also another infection that is a yearly infection in the US caused by West Nile virus. So you have West Nile encephalitis. A lot of people get infected with West Nile virus, but very few people suffer severe consequences from it. About only 1% or so would have serious fetal neurological illnesses where it will destroy their their nervous system. Sorry. The reservoir is birds. We have it now in almost every state in the country. They are transmitted by mosquitoes, mainly the Culex mosquitoes that transmit West Nile virus. So in all these states, in California, you have all these mosquito districts in various counties where they yearly they take measures to protect the residents from West Nile virus. Then you have bovine spongiform encephalopathy, which is also called mad cow disease. We've seen increases in that since the 70s. There's been outbreaks in the UK in the 80s and the 90s as well. It's caused by what we call prion. Prions are diseased amino acid disease proteins. And so, for instance, this is a normal prion proteins. There are proteins in the body that are called prions. Now, if these become diseased because of how they are folded, because this folding is different from this folding, because of how they are folded, they become diseased. They, in turn, could also infect other prions that exist in the body. And it could result in fatal, it almost always results in fatal neurodegenerative diseases. And so if you have a cattle that is suffering from such a disease and it's eaten by humans, because the meat is infected, the human would also become infected with a prion, and then it will spread to other prion proteins within the human. Another example is the Creutzfeldt-Jakob disease. What we've seen is that in areas where there is bovine spongiform encephalopathy or mad cow disease, you also see incidences of Creutzfeldt-Jakob disease. So the two are kind of going together. There isn't any evidence of person-to-person transmission, but it also results in a neurodegenerative disease in the infected humans. Then you have E. coli. E. coli, the 0157 H7 strain. It produces a toxin called Shiga, Shiga, the Shiga toxin. And it has resulted in certain outbreaks in people who have eaten unwashed vegetables that are contaminated or undercooked meat or raw milk. And it may cause even severe kidney failure or even death. There isn't any evidence that antibiotics are able to help, but mainly people hydrate a lot, and there is a lot of fluid replacement, and you may recover from it. There was also outbreaks of this same strain in 2015 in Chipotle restaurants. We have 60 cases across 22 states, and some people, and more people getting hospitalized. Those of us in California have suffered from some of these outbreaks, and so it's close to home. Then you have viruses. You have Ebola hemorrhagic virus, caused by the Ebola virus, and some of the symptoms is that it causes internal bleeding. So there is hemorrhaging and clotting within the host. Outbreaks happen every now and then. However, because it kills the host very quickly, and the transmission rate is not that serious. And so if you're killing your host very, very quickly, then your host is not going to be spreading it to as many people as otherwise would have happened. And so we had an outbreak in 2014, 2016, and there was some importation into the US. Currently, we're doing better in terms of controlling this particular virus. There has been plasma treatment that's worked, and there are vaccines that have been trialed and found to be successful so far. HIV has been with us since 1981. It continues to be with us. The good news with HIV is that with advancements in treatment, it's now more of a chronic infection than the death sentence that it previously was to those who got infected. You have Cryptosporidiosis, which is caused by protozoa, causes 30% of diarrheal diseases in developing countries, and it's also a major cause of childhood diarrhea and childhood malnutrition. There are cases here in the United States, and you have a number of cases on the increase, especially during summer, when a lot of people interact with water bodies. Then in 2015, not too long ago, we had an outbreak. Zika was spread by mosquitoes. The main issue was that infections could be transmitted to newborns, and that there could also be sexual transmission. They are transmitted by mosquito vectors, the Aedes aegypti and Aedes albopictus mosquitoes. That's the main cause of transmission. There are other forms of transmission accidentally in the lab or through blood transfusion or organ transplant, but those are not the main route of the transmission. We still don't have a vaccine for it. The fetus of mothers who suffered from Zika were likely to suffer from microcephaly. So that was the risk that was attached to that particular virus. And then we have COVID-19 disease caused by COVID SARS virus 2. And it's originated in Wuhan, China, in December 2019. We still continue to battle it today, and the reason for which we are having a class asynchronously online. And symptoms include cough that persists, shortness of breath, and fever or chills. There is fatigue, muscle or body aches, a new loss of taste or smell. There is sore throat, running nose or congestion, nausea and vomit, and the list goes on and on. But these are the most common ones, and we continue to learn every day about this particular virus. And for prevention, we know that social distancing or physical distancing works. Wearing a mask when you cannot maintain such distance, using alcohol-based disinfectants, washing your hands regularly and properly, and the like. So that brings us to the end of the third part of our presentation of our lecture on Introduction to Microbiology and Microscopy.